Okay. Okay, can you see my slides?
Yes.
And I want to do one more check whether the slides are changing. Is it changing or?
No, now it's not changing.
Okay, so I have to reverse the order in which I do this. Okay, so let me stop sharing and. Okay, so I have to present first and then share. Now let me share. Okay, can you see my slides?
I can see your slide.
Okay, then let me check. Now it's changing.
Yeah.
Okay,
Yes.
perfect.
Yes.
All right. Okay.
So probably let's wait a few more minutes to give more people a chance to join us, especially those in the US. Still early for them.
Oh yeah. There is a rainstorm happening here, so there's a possibility I can lose power. Okay, so just kind of to one, there are power failures in the area. So if I vanish in between the talk, it can be due to a power failure.
Okay, I think we can start introducing our speaker. So, good morning or good afternoon or good evening, everyone, depends where you are. I'm Amir Ali, the coordinator of the CRNS talk series, a lecturer at Plymouth University in AI and robotics. And today is my pleasure to have Dr. Dileep George from Google DeepMind US. Dr. Dileep George has his master's and PhD in electrical engineering from Stanford University. He's a scientist and engineer working on AI robotics neuroscience. He co-founded 2 companies in AI, Namenta and Vicarious. At Namenta, he co-developed the theory of hierarchical temporal memory with Jeff Hawkins, and at Vicarious, he was a pioneer of AI robotics and developed neuroscience-inspired models for vision mapping and concept learning. Vicarious later on was acquired by Alphabet Incorporation to accelerate their efforts in robotics and artificial general intelligence. And as a part of this acquisition, Dr. Dileep George and his team of researchers joined DeepMind, where he's currently developing and working. So today he will be showing us one of his research studies that he published recently, with the title of Reverse Engineering Cortical Microcircuit Models for Visual Perception. Dr. George, it's over to you.
Thank you, Amir. Thanks a lot for inviting me to give this talk. It's great to be here. And so the topic of the talk is reverse engineering cortical microcircuits of vision. So we all know that we all have a cortex, and there is structure within the cortex, and different part of the cortical sheets look roughly the same, but there's detailed micro architecture within them. And one of my goals is to be able to understand what these microcircuits do and what the computations within that are. These are typically referred to as canonical cortical circuits because there is a prototypical organization of these circuits within different regions and across regions. And there is it's known that these are organized as cortical columns and laminae. So there is structure there, and there's a prototypical connectivity of how these cortical columns interact with the thalamus in every region, and also through the hippocampus. So this is why it's known as the canonical cortical circuits. And typically, neuroscientists anatomists produce diagrams like the ones on the left or on the right. On the left, it's a very detailed diagram of all the data that we have accumulated from neuroscience, neurophysiology, and anatomy about how neurons are organized in these cortical layers. So they would say, oh, each cortical region has maybe 6 layers, and there are neurons in these different layers, and this is how they are connected. This is how they interact. If you poke a neuron here, this neuron will light up, etc., etc. That's on the left. And on the right, you typically see summary diagrams, which are reduced schematic versions of the diagram on the left, showing, oh, this is the primary circuit, and this is how the dominant interactions are. However, both these descriptions are not computational in the sense that you cannot take this circuit and make it do something. The circuits don't work, so they cannot be a computational description. It's more of a summary diagram. What I am interested in is having a cortical circuit model which is also computational, that it has to do something that the brain does. So how do we go about that? If you do bottom-up studies, they give very important clues, but the computational model is too local. And if you go from the connectome, where you have all the connectivity information to whatever detail you are recording it at, it's not very informative without connecting it to neurophysiology and behavior. And simulating the connectome itself is unlikely to give any computational insights. So instead of doing I hope
excuse me.
I hope you can hear, and there's no problem. There was an interruption, so I thought there was a question.
Interruption.
Okay. I'll just continue. Okay, great. Okay, so how do we reverse engineer the brain and to build AGI and to understand how the brain works? And the answer that I am pursuing is that tackle both at the same time. And there's a particular recipe I'm going to I'm following, which I'm going to explain in the next few slides. So this recipe is to consider 3 things at the same time. So you can think of these are 3 corners of a triangle. So you consider the brain as one corner, the world as another corner, and the computational model as the third corner. And of course, from the brain, we have lots of information about the neurophysiology, the neuroanatomy. There's cognitive science, which describes what the brain is doing. And on the other corner, we have the physics of the world and real-world test sets. And the brain is organized in a particular way because the world is organized in a particular way, because brain is trying to model the world. So any organizational property that we find in the brain has a counterpart in the real world. And so if we can connect the property of organization of the brain with a property of the world, and also explain it from a computational principle, which is the third corner, then we have found something useful, because there are so many details about the brain that might not be about computation. Some of them can be just about just being able to maintain the neural circuits over time, or just about staying alive, or cleaning up stuff. So all those details might not be important for computation. So to understand something that is relevant for information processing as an organizational principle from the brain, we need to be able to connect the dots of how it connects to property of the world, and also how explain it from a computational angle. And so the first part of that is being able to build a computational model which is derived from the brain, but can stand on its own. And this was the first part that we did. This was a paper that we published in 2017, a generative model that trains with high data efficiency and breaks text-based captchas. So that model incorporated information from the brain, which I will talk about, and also it had test sets from the real world, and so it can stand on its own. It doesn't need to refer to the brain to describe it. It's a purely computational model, although it incorporates information from there. And a few properties of this model was in relation to the current deep learning models are: one, it emphasized compositionality and controllability top-down, using top-down attention. And it was, at that time when it was published, about 300x more data efficient than comparable deep convolutional neural networks on similar tasks. And the second step, which is what I'm going to spend most of the time today on, is to take that computational model and compare it with the neurophysiology and anatomy, and derive a detailed microcircuit model on how the computations in that model is performed in biology. And that's what I'll be spending most of the time today on. But of course, it requires some understanding of the computational model, so I'll spend some time on that as well. Okay, so this first part is just explaining what that computational model is at a reasonably high level. So when we published this computational model, we did get quite a bit of attention, because it broke captchas. The captchas that you used to fill in at that time, which was text-based captchas, was fundamentally broken by this paper. And that's why we got the attention. But that was not the reason why the attention was not the reason why we chose captchas as the problem to tackle. We chose it as a problem to tackle because captchas exemplify the strong generalization we seek in our models. So, for example, I can show you these very diverse set of captchas. You might not have seen many of them, and you haven't been trained on many of them, but you can parse all of those captchas. In fact, a 5-year-old child should be able to parse all these captchas without getting any training examples from those particular style. And I can keep creating new styles that anybody would be able to parse. And that's the data efficiency that humans have, that current models are still not there yet. And it doesn't even need to go to strings of characters. You can even just do it for a single character, and I can make the same statement. Another aspect of the generative model that I want to emphasize in relation to existing more popular deep learning models, deep generative models, are in most pervasive deep learning models that exist today, the encoder and decoder of the generative model are separate pathways. The encoder does an amortized forward pass, and the decoder does a backward pass for reconstruction, but these are two separate streams. And that's because these models are they have structure only at the stochastic variables, which is at the very top of the model, and also at the bottom of the model. And everything in between is a deep neural network without any structure within that. So there are no stochastic variables at every level. So there is only stochastic variable at the very top of the network. And at the very bottom of the network. And so that means these two streams cannot interact, and you keep them separate, which is very different from how it is in the brain. And also having these separate have computational implications, computational shortcomings, which can limit how the kind of tasks that they can solve. Whereas in RCN, we have stochastic variables at every level in the network, and the forward and backward streams interact at every level in the hierarchy. And this is something that we took on as something that will be needed in the future for models. As these models become, as we build the next version of these deep generative models, this interaction between variables at every level will be needed, and we will need models that can have stochastic variables at every level, because we don't want to just do classification. We also want to do reasoning and mental simulation, all using the visual cortex. And for doing that, we need stochastic variables that can be controlled at every level in the hierarchy. In addition to that, we also emphasized lots of properties from biology. I will give a table here, but I will highlight only a few of them. So there's a biological feature, there's a computational algorithmic reason, and how it is represented in RCN. Let me pick up one arbitrary row here. For example, object-based attention is a property that biology exhibits. And the reason why it needs to exist in the model is that it gives you compositionality, so that you don't have to train on combinations of objects. You can train on single objects and still be able to parse scenes that have multiple objects in them and be able to manipulate mentally each object independently. And in RCN, this manifests as having only positive weights and object and background factorization. That's how this property manifests in the model. And many of these properties are related to each other. They don't need to have, they're not separate things. Object-based attention and hierarchy, for example, are related. And feedback connections are related to how inference happens in the model, that inference cannot be just a single forward pass, because these objects are modeled as separate entities that need to interact. And that interaction can be, since you do not know where the objects are upfront, you need to first have a hypothesis on where these objects are, and then they will interact in the hierarchy and try to produce explaining away. So object-based attention and feedback connections are connected to each other. They need to be compatible with each other. And that is the challenge of building a model like this. So now I will go through a few of these and explain it in more detail. So the things I want to focus on in this talk are a few features of the model. One is factorized contour and surface representations. Second is lateral connections between contour features. And the third is object-based attention and explaining away. So factorized contour surface representations, you can also call it shape and appearance factorization. So in behavior, we know that we can recognize objects with arbitrary shape and appearance combinations. You can recognize strawberry banana without being trained on that, or sorry, strawberry elephant. You can recognize rainbow bananas or chair made of ice. These are all unusual objects whose shape and appearance combinations that we haven't seen before that we are able to recognize. And there is evidence in neurophysiology that there are concurrent processing streams in the visual cortex, one related to shape and other related to contours. Sorry, one related to contours and other related to surfaces. So contours encode the shape, and the surfaces encode the appearance or even the surface normals, etc. And this separation exists very early, like at birth. It's not a learned separation. It exists at birth. And also there is separate dynamics for processing texture versus contours. This is all evidence that we see from biology. And what might be the computational principle? So I'm looking at the three corners of a triangle. What might be the computational principle behind this? And the computational principle behind this is that the natural world has two levels of smoothness. So natural world is typically piecewise continuous, And the discontinuity itself is piecewise continuous. So images have regions of piecewise continuity, and the discontinuity is at the contours, and the contours themselves are piecewise continuous. So that property could be thought of as a factorization of shape and appearance. And it's not applicable just to images. It can be applicable to videos, flow fields, et cetera, et cetera. And you can see if this computational principle is not used in a model, then that model will be able to recognize unnatural images. For example, QR codes. The model will be able to classify QR codes at very high rate, which is what you can do with convolutional neural networks, for example. But human visual system is not very good at classifying QR codes. That's because we have this assumption about smoothness of natural signals embedded in our visual system, which makes it very hard for us to parse or understand QR codes, but it makes it much more efficient in dealing with natural signals. So this is where the that trade-off comes in. You can put in an assumption that will make your model very good at some set of data, in this case, natural signals, and it will make it less likely to model this unnatural data, which is the QR codes. And how does this appear in our model? It appears in our model as an interaction between contours and surfaces. I will probably have another slide which explains it in detail. Okay, so here, so you can think of RCN at every level in the hierarchy, although in this paper we did it only at the bottommost level, having two sets of nodes. One set of nodes correspond to the surface features, another set of nodes correspond to the contour features, and they interact in a particular way. So the surface features interact saying that, oh, if I see a surface in one place, a surface in a nearby place should have the same appearance or the same surface angle. So that's kind of a surface continuity criteria that this factor graph encodes. Except when does this surface continuity criteria break? It's when there is a contour in between that. So this is a three-way interaction between surface features and contours. Surfaces are continuous if there is no contour in between. This is the information that is encoded in the factor graph. And if you sample from this factor graph, it will create, for example, if you clamp where the contours are and generate from this factor graph, it will create cubes. In this, because in this particular case, we clamped these particular nodes as contours, the green nodes as contours, and the blue nodes as inner contours. And if you generate from that, it will generate different surface appearances, just showing how the factor graph works. The other property that I want to emphasize is lateral connection between contour features. So the behavior that we see is that even when the contours are disconnected, we infer a continuous flow of the contours. And that's because in the visual system, we have lateral connections between contour features that emphasize contour continuity. And people have studied this, and the properties of this are computational properties behind this are summarized, for example, in this paper by Lawler and Zucker. And in RCN, it appears as lateral connections between contour features at every level in the hierarchy. So I mentioned that RCN, which is the name of our model, which probably I forgot to mention earlier, it stands for Recursive Vertical Network. And RCN has features at every level in the hierarchy, features and pools at every level in the hierarchy. And the contour continuity connections exist as lateral connections between the contour features at every level. So here is an example of how RCN has a two-level RCN model, which would have, for example, features corresponding to a line segment here and a line segment here, and they occur in the hierarchy. So the higher level feature is encoding this corner, and these are all learned connections. This is not hard-coded. These are all learned connections, but this is just, I'm explaining it in a simple form to make it understandable. So this higher level feature encodes this so this red feature encodes this corner, and it can be broken up into smaller features at the lower level. And these green things that I've highlighted, those are the factors that encode the compatibility between contour features here and contour features here. So if you have this model, and if you turn off these lateral connections, if you do not have these lateral connections, when you generate from the model, it would create corners which are kind of jagged. They will look disconnected because the generation on one side is uncoordinated with the generation on the other side. Whereas if you have the lateral connections, it would generate contours which are, or these corners which are smoother in variation. And this is something that you can control also in the hierarchy, which okay, I probably will have a slide next. So, and these contour lateral connections are such that they encode higher order interactions. So for example, if you look at a feature, this is a contour feature. And if you look at the different curves that can pass through that contour feature, so you can see that curves of different curvature can pass through that contour feature. If you encode these interactions using only first order connections or first order Markov model, then you will mix up contours which are flowing in one direction with contours which are flowing in other direction, which might not be a property that you want to have because you want to keep long distance connections. So what we do in our model, and this is a property of the model for encoding higher order interactions, so we keep multiple copies of these contour features. These are all contour features which talk about the same thing locally, but they are connected differently for different curvatures of contours passing through that. So these contour features are all talking about the same set of pixels in the input. And so looking bottom up, they will be talking about the same contour features, but they are all in their lateral connection, they are talking to different features in their neighborhood. And this sort of connectivity, where we call these as clones of the same contour feature, they are connected to different things. So basically these contour features differentiate because of their lateral connections. And this is a property that we repeat throughout the model to encode higher order interactions. And so this is typically what a model will look like for a letter. If you pull out an object's connections from the model, this is typically how it will look like. Of course, this requires very detailed surgery within the model. The model will have multiple objects sharing features at different levels, and it looks much messier, but you can, because you can do top-down attention, you can pull out the model corresponding to an object for a particular evidence that is presented. But in the model, all the objects interact all the time, etc. Oh, and I wanted to show that these lateral connections that I mentioned can be controlled at every level. So if you turn off the lateral connections at the bottom, then what happens is that you will create very jagged local edges. So for example, the third column here, that's an example of turning off the lateral connections at the very bottom level. You can, if you do it at an intermediate level, you will keep local structure, but the global locally things will be smooth, but globally things will be jagged. That's the last column. And so you can control the shape variation using these lateral connections at multiple levels in the hierarchy, and they produce variations at different structural levels. Okay, and the third one is object-based attention and explaining away. So in behavior, we can see that we can separate objects even when they are overlapped. The two transparent objects overlapping in the same area of space, we can separate them. We can say, oh, this belongs to the face, this belongs to the house. So it's not just based on objects being separated in space. We can do that even when they are overlapping. And our models do have this property. Once you have this property, then you also need to, along with it, need to have this property of explaining away. So let me explain what I mean by that. So if I give you a CAPTCHA string and I show you locally, this is the evidence, you might think that this evidence belongs to the letter M. And if I just expand that window, that again looks like an M. Now you probably are even more sure it's an M but you are not sure anymore. And if you look at the whole scene, it's not an M because it's U, a combination of U and N explains that evidence better than an M locally. But you can, if you look locally, it looks like an M, but globally it's not an M. So so this is the effect of explaining away. Local evidence can support hypotheses that will get explained away when you look at the longer context. And the brain has to do that because it's encoding each object separately and being able to interact between them is an important property. And the way it happens in the model is that if you give a complicated scene, if you do a first forward pass, it will bring up object hypothesis at the top. But some of those object hypotheses can be hallucinations of things that there is evidence for it, but the evidence is explained away by other pieces of evidence. For example, if you do a forward pass here, it might come up with a hypothesis V, which exists in the input. You can see that there is a V in the input. There is evidence for it, but it's not the right explanation because it's explained by A and K being present there. So this is what needs to happen during the backward pass, so that these are explained away during backward pass and the further forward pass. Okay, now how did the model do? It did very well on CAPTCHA parsing of different kinds. And as I mentioned, it's not trained on the examples from these CAPTCHAs. We train on clean font examples, and it generalizes to different deformations and clutter without specific training. And you can do a direct comparison with deep neural network models. So our model was trained on 260 character images. A comparable deep neural network model required about 500,000 CAPTCHA strings multiplied by 8. That's the number of images it saw to get to the comparable accuracy. But then if you test based on increased spacing between the characters, which actually makes the problem easier, our model will recognize roughly the same or give a slight improvement. But a deep neural network which was trained on only a particular spacing would have its accuracy dramatically go down if you test it on increased spacing. That's because the neural network was trained on the combination of things. It needed to see how these characters occur with each other in combination to be able to parse the scene, whereas our model, by using explaining away, was able to do it independently for each object. And that's the reason for the robustness there. And another thing I want to emphasize is that RCN is a model that can do recognition, segmentation, occlusion reasoning, all in the same model. So it can do recognition, reconstruction, parsing of the scene, and it's all the same, and occlusion reasoning, and just unconditional generation, all in the same model. These are all, as I mentioned, there is no separate encoder-decoder network. You have one network on which you can ask different questions, and these are all different queries on the same model rather than training different models for doing different things. So it's an integrated vision model where you can do classification, parsing, reconstruction, generation, etc. Whereas you typically have to train a different model for each of these tasks. I mean, it's definitely changing. This is, you have to take this as a statement from a few years ago. Okay, now coming to how does this all convert to cortical microcircuits? So now we are taking everything that I explained so far is a purely computational model, although it uses inspiration and information from the brain. But now we can say how do the computations in that model correspond to computations that happen in biology, and try to fix that. So so you can think of it as a jigsaw puzzle, solving a jigsaw puzzle. So what biology gives is information about computations that can happen in different layers of the cortex. There are constraints. If you do this computation here, the anatomy gives you constraints on whether that computation is possible in a particular layer or not. Some constraints, some computations might be possible in any layer, but some computations might not be possible. So you have to kind of keep these pieces around and see how does it all fit together. And that's why it's like solving a jigsaw puzzle. And there can be multiple solutions. And this is not going to be a unique one. There can be multiple ones, but at least one unique one is good to understand, or one solution is good to understand. So the kind of questions that we are interested in is, for example, what's the function of a cortical column? What do neurons in different laminae do? Which connections in a column are innate? What computational role might that have? What are the computational roles of blobs and interblobs? Blobs are cortical columns which correspond to surfaces, and interblobs are cortical columns that correspond to contours. And what do feed-forward, feedback, and lateral connections contribute to an object percept? So these are examples of questions that we are interested in from the context of biology. And what computations are performed in the dendrites. These are examples. So 2 steps in deriving this cortical microcircuits. So the one is to take the factor graph message. So I mentioned that the model is specified purely computationally, and it is specified using a factor graph, which is a way to encode probabilistic graphical models. And inference in this factor graph is done using message passing, which is just a local algorithm where each node tells its neighbor what does it believe in, and those messages are exchanged. So it's a local gossip algorithm, very much like what can happen in the brain in terms of the locality of the computation. So the first step is converting this factor graph to a biological neural circuit to say, oh, how can this factor graph computations be implemented in what looks like neurons, or with those neuronal constraints, and then taking that biological neural circuit and mapping it to the different anatomical and physiological constraints. Because this biological neural circuit just says, oh, this is implemented in neurons. Those neurons can be placed anywhere as long as the connections remain the same. But anatomy gives us constraints on where those neurons can be placed, because there are constraints on, oh, these are the connections and how they are connected within a column, across lamina, et cetera. So these 2 steps, converting to biological neural circuit and placing it in the cortical anatomy, is what's involved in deriving this microcircuit. Okay, so I'm going to show this for a very simple graphical model, because the RCN graphical model has many pieces in it, and it's very complex. But if you understand it for a simple graphical model, you can replicate that process for the more complex one too. And the good thing is that the small piece that I'm explaining is still part of the more complex graphical model of RCN. So it's not wasted. So the very simple graphical model I'm explaining here is this one, where you have a set of features at the top and set of pixels at the bottom. And what this graphical model is encoding is that, oh, this feature A, if I turn on the feature A, it turns on these 2 pixels, the left 2 pixels. If I turn on feature B, it will turn on all 3 pixels. And if I turn on feature C, it will turn on the right 2 pixels. That's what so you can think of this graphical model as encoding the feature descriptions and the interactions between them. So what happens if A and B are on? If A and B are on, it just makes it more likely to, if 2 parents are on, it just makes it more likely to turn on that particular feature. So it's an OR interaction between these parents. So that's what the graphical model encodes. So the multi-parent interaction here is an OR interaction, or a noisy OR interaction. Now, so that's the graphical model. This is the corresponding factor graph. That's just a different way of representing this same information. So these nodes became these nodes here. There are these factors of their interaction, basically saying, oh, if A is on and B is on, this child is on. That factor which encodes that information is explicitly called out here. This is what I meant by an OR interaction. But these are encoding the same information. Now, the question is on how do you convert this to a biological implementation? So before that, let me show you some property in this model, which is the explaining everything that happened I mentioned earlier. Suppose all three pixels are on in the input. This is, how do we do inference in this model? Like, suppose all three pixels are on, the first forward pass, this is the message passing pass, the first forward pass through this model will turn on under some conditions, all three features at the top, because locally there is evidence for the presence of either of these features. So A, B, C, all are present in the input. Now, when you do backward pass and a further forward pass, there will be competition between these features because of explaining away at each of these nodes. This higher level feature will say, oh, I want to explain these ones. So it should take away evidence from this one. So there will be an inhibition in the sense that this forward evidence will not be passed fully to this node. It will be taken up by this node, and that's the explaining of interaction that happens between in a graphical model. Now, how is this computation? This is a graphical, extremely simple computation. How will that get implemented in neural circuits? The first thing to keep in mind is that neurons can pass messages only in one direction. You cannot. So here is a node in a factor graph, and you can see that there are multiple messages coming in and multiple messages going out. That clearly cannot be one neuron, because neurons can pass messages only in one direction. So even to implement this simple node, which is B here, you need multiple neurons, because neurons can pass messages only in one direction. So you need a neuron for sending out each of these different messages that is going out of the node is being sent out by a different neuron. And similarly, for receiving messages also, you might need different neurons and then combine. So even a single node here expands into a bunch of neurons, because neurons are unidirectional. And similarly, this factor here, which I mentioned, which is an OR factor, and there is explaining away computation happening here, that expands into multiple neurons here, again, because these messages need to be kept separate for different parents, et cetera, et cetera. And then you can even see a particular computation. What is the computation that needs to happen in that neuron to do this explaining away thing? And also what is, and you can go one more level detail to see what is the interaction that happens, what's the physiological interaction that happens in this neuron when I change top-down signals to that neuron versus bottom-up. And you can produce a transfer function of facilitatory and inhibitory effects of top-down signals, all from this very simple factor graph. So that's just showing that taking a factor graph and converting it into a biological implementation has a thought process behind it, and it is mostly guided by computations in this computation in neurons can be only unidirectional, and also neurons are either excitatory or inhibitory, et cetera, et cetera. So those considerations is what determine how a factor graph gets converted to a biological circuit. But keep in mind that that implementation is not unique. There can be multiple instantiations. And we do consider efficiency as another factor. There are all ways of converting these computations is not equally efficient. And efficiency of connections and computations is another thing that comes in the into the picture. Okay, so this is, this was a, what I explained earlier was a very simple graphical model, but RCN graphical model is more complex, because that piece appears at every level. There are features, lateral connections, pools, etc. And so this is the detailed factor graph of one level of RCN just for the contour features. So so we have features, laterals, and pools, as I mentioned earlier. And you can see that the piece I explained earlier, which is having features interact with lower level evidence, appears here. And then there are lateral connections between copies of those features, which is what I had mentioned earlier. You need to keep these different copies around, because those copies help you model higher order interactions. And then there is pooling on top of that. The pooling is more like pooling in a convolutional neural network. And then that's the message that is passed up. So all these components exist, and so we need to consider all of them at the same time. And if you take a vertical slice through this one, let's say this would correspond to a piece of RCN graphical model that represents a patch of the input, visual input. And so you can think of that as a macro columns graphical model. And so each of the nodes in this graphical model and each of these factors have computations associated with them. You can say if these are the messages coming in to this factor or a node, what are the messages going out, and how is the computation derived? So that's the message passing computation, and so that's what is explained here. This is all purely from the graphical model. And then, as I mentioned, for each of these factors and nodes, you can see what is the biological implementation. And so now I'm going to talk about the biological neural circuit and how we map it to anatomical and physiological constraint. And this is where it's a jigsaw puzzle solving problem, where because there are anatomical constraints, because, for example, if you know that you are placing a particular neuron, a particular computation in layer 4, that imposes some constraints on where its subsequent computations can be placed, because layer 4 only connects to layer 2, 3 in the feed forward, and also layer 5, so it gives you a set of constraints on where the subsequent computation can be placed. So all these different factors and computations and nodes that you have derived can be placed in anatomy in only some set of combinations, and that's what we do, and that's a manual process. There is no automation there, and I just want to highlight the important pieces of how that is done. One is to realize that the factor graph description entangles the feed-forward computation and feedback computation and lateral computation all in the same thing, because these nodes are bidirectional. So in a biological implementation, that is not possible. So you have to you can think of having 2 copies of the same set of nodes and factors, one for feed-forward computation and another for feedback computation. You can see that I have just copied this same thing here. This is just for convenience. And then these computations are so features, laterals, and pools map to different layers in the cortical laminae. So you can say the feed-forward computations here of features map to layer 4, the pool computations map to layer 2, 3, and layer 1 is a broadcast layer. And similarly, the same pool computation, but now incorporating not just feed-forward, feedback information as well, and these need to be kept separate for this message passing to work. They happen in layer 5, and then in layer 6 for this feedback is unfolded and sent to the children. So the important takeaway from this picture should be that you need to keep 2 sets of computations that are talking about the same things, but one for feed-forward, another for feedback. And you need to do that because neurons have only a single direction of communication. And it's a very complex circuit, but I want to focus on a few takeaway messages that you can hang on to. One is treating cortical column as a random variable, and I'll mention clonal neurons in the cortical column. The other is the idea of thalamus as doing explaining away and gating. And then I will focus on some virtual neurophysiology we can do with these models. So cortical column as a binary random variable. So you can think of, in general, a cortical column as signaling the presence and absence of a feature or a concept. So for example, this cortical column can be representing this contour segment. And nearby cortical columns might represent different contour segments, and they might all be talking about the same visual input patch, which corresponds to a macro column. But each one is representing a feature or a concept, and all the computations within that cortical column are just to figure out whether that cortical column should be on or off. So the goal of the cortical column is to compute its belief that it's part of the most likely explanation for the evidence. So when an evidence comes in, which is a picture comes in, all the cortical columns are trying to compute the overall belief. Oh, is this a dog in the picture? Is there a cat in the picture? And which contours of the picture are on, etc. Depending on where we are paying attention to. So it's trying to come to this distributed belief, and each cortical column is trying to compute its role in that distributed belief, seeing whether am I part of the distributed belief or not? Am I on in this overall thing that we are paying attention to or not? That's a map inference. So that's the role of a cortical column. And the different layers of the cortical column encode different aspects of this random variable. So if I'm thinking about this feature, which is a horizontal line feature, the different layers are encoding different aspects of this random variable. What are these different aspects? So layer 4 encodes the feed 4 representation of this feature, which is saying, oh, if there is there bottom-up evidence for this feature? Layer 2, 3 is encoding the participation of this feature in different contour continuations. So if is my neighbor on? Does my contour continuous neighbor give me any evidence of me being on? Layer 2, 3, another set of neurons are encoding in a temporal succession, am I part of a sequence? And the pools are encoding a community of these things. And then layer 5 is encoding the same lateral continuation, but again with feedback from above. Layer 5b is computing the belief of this feature. That's where finally everything comes together to see whether I am on or not. And layer 6 is the feedback computation. So the important thing to keep in mind is that all these computations are talking about the same feature. It's just different pieces of evidence that support whether this feature is on or not. And what are those different pieces of evidence? Layer 4 is feed forward. Layer 2, 3 is simultaneous lateral context. Layer 2, 3 is temporal context. Layer 2 is pooling and community. Layer 1 is information from above, hierarchical information from above. And layer 5b is the belief of this feature. So this is what I mentioned. All the neurons in the cortical column are talking about the same feature, but the different aspects of that same feature and how they all come together to determine whether the feature is on or not. And this picture also gives you some idea on which connections within this cortical column can be innate and which need to be learned. Most of the connections within this cortical column across the laminae can be hardwired upfront because this participation is already determined. It's basically that's something evolution has figured out. And then because, for example, the lateral context is encoded in the connections between the columns, not within the column. Within the column, most of the connections are about how to combine the beliefs given these connections already exist. So they can be populated upfront. And there is some evidence to support this one. So so what I'm saying is that excitatory neurons in the different laminae of the same column share the same bottom-up input. And what differentiates them is their intercolumnar connections. And there is this idea of clonal neurons in a column, basically saying that all these neurons in a cortical column originate from the same progenitor during the ontogeny. Then they differentiate only through lateral connections. So for example, this paper, which is a fairly recent paper, strongly supports this idea. Okay, now coming to thalamus for explaining away and gating. I mentioned that you need to do explaining away between objects or features because each feature or object is encoding just the local evidence and their interactions are figured out on the fly during inference. And when we did this mapping on how to take the computations in RCN and map it to cortical laminae, there was no good place to put this explaining away connections So you mentioned this, the explaining away connections are happening here. This is the fragment of the graphical model that I explained earlier, where the explaining away connections happen, interactions happen. And the only way we could fit in was by putting that explaining away connections into the thalamus. And that is actually pretty pleasing, because there is, when two levels in the hierarchy interact in a visual hierarchy, they always go through the thalamus, and there is a particular form of connectivity between them. There are two forward pathways. One is direct cortical, and another is through the thalamus. And what our model suggests is that thalamus is doing this explaining away and gating. For example, this particular factor that I mentioned earlier, where there are multiple higher level features trying to explain the same pixel, they need to exchange information. That particular computation would fit the circuits of the thalamus. And not only that, this is also the same computation that needs to happen if you want to hold attention to a particular object. So, which is basically saying, I want to say, oh, I want to pay attention to B, which is saying I turn on feature B top down, and that should turn off all the other features which are supposed to be explained away. So any kind of gating that you want to do based on top-down attention can be implemented in the same circuit where this explaining away happens. So that makes sense to put that in a separate nucleus where you also have other interactions coming in. So that's our explanation for the computations happening in thalamus. And it's very pleasing that people have actually done detailed study of how circuits work in the thalamus, and they have even investigated between alternatives. And the model that comes out of RCN is compatible with the alternative anatomists and physiologists have found as implemented in the thalamus. Okay, so I will skip this slide because I did go through the detail of this before. The thing I want to mention from was that this is also, if you look at the properties of neurons, the network of neurons, it's not a property of a single neuron. A neuron is connected in a particular way inside the thalamus. If you look at the physiological property that implies how does the bottom-up information get gated based on top-down being on or off, people have studied that, and what comes out of that is roughly compatible with what comes out of our model. So this one I will skip. So let me focus on virtual neurophysiology for the last few minutes. So we know that visual cortex humans can exhibit the subjective contour effect, where we can see contours where contours are not there. For example, we see a triangle here, and we kind of hallucinate contours in between to explain that triangle. RCN can do that. RCN will do that too, in the sense that if you give this triangle as input to RCN, without ever having being trained on that one, it would classify it as a triangle. And if I stay my attention to triangle, it will turn on these features, which are these magenta nodes. It will turn it on, and those are the nodes for which there is no bottom-up evidence. We are completely hallucinating those nodes, and this is a property that we can see in biology, and people explain it. It's now, since we have this virtual cortical circuit, we can simulate in detail in our virtual cortical circuit and see what is the temporal dynamics of this happening, and in which layers will these computations happen, and which layer will settle first, which layer will settle next, etc. So we can predict the detailed hierarchical and laminar dynamics of subjective contour effects using this model without ever. The model is not constructed to explain this. This is an emergent phenomena from the model. The other one is neon color spreading. I have to mention both these were inspired by work that Steve Grossberg has done earlier. Both of these effects are emphasized in his work. And his models can do this too. It's just that our models can it's produced a different explanation, more from a graphical models perspective, explaining away perspective, and also, of course, keep in mind that our models can also do captcha parsing and parsing of objects in the scene in the real world, etc. So and so that additional constraint that it needs to do real world parsing makes some aspects of the model more detailed and more real world tested. Okay, so neon color spreading is an example where our visual system is doing two levels of hallucination, where there are the virtual the subjective contours are hallucinated because there is no real contour for this circle here. All the contours are subjective, except for at the very boundary between the gray and the black here. And then the surface inside, you might be seeing this as a gray color, but it's actually white color. So the color here is also hallucinated. So let me see whether so the boundary that you're seeing is hallucinated, and the color internally, which you might say is gray, is actually the same as the background color. So the color is also hallucinated, and it's cool that the hallucinated color doesn't leak out of the hallucinated boundary. The color stays within the boundary. So that's what is cool about this illusion. And this same thing will happen in the RCN model too. Again, keep in mind that the model is not constructed to explain any of these things. These are all just side effects of the contour and surface factorization and object-based attention kind of things that are more fundamental in the model. And these are just side effects of that. So what happens is that if you put this as the stimulus at the top, it will recognize the circle because there is local evidence for the different contour pieces, and it will all stay together. It detects the circle just like the subjective contour effect in the previous model, in the previous slide. And then it would clamp those contour features as on, which would restrict how the surface features can communicate between each other, which I mentioned in one earlier slide, where contours and surfaces have a three-way interaction. If the contours are on, that will constrict the surface continuity propagation. So the interaction between these two things, the contours and surfaces, will produce this subjective contour effect, where it fills in the color in the interior as the color at the boundaries, because in a contour continuity the surface continuity is enforced as part of the model if there are no contours intervening. So that's the explanation for neon color spreading. Of course, we have, there are RCN is not the final answer. The perception is still a unsolved problem. And one of the things that I want to pull out as not solved in anywhere is the idea of objectness. Humans can deal with completely new objects without any problem. Where, for example, the objects in panel A here, entirely novel objects. There are no names for them. Humans have no problem saying, oh, which are the objects, where are the boundaries, can I pull this object separately from the other, etc., etc. Currently, none of our systems can do that. RCN cannot do that. We have made an attempt at doing that. That's just a partial. It works in some settings, but definitely not. So this is an unsolved problem. So perception in general is a very unsolved problem, and we are making progress towards that, and that's the next thing that we are working on. So hopefully the talk explains this triangulation strategy to give you a rough idea on how we are thinking about all these different pieces. You can find more information on my website. There are other papers on that website which is connected to this one, and these are a few other papers that are related. And recently we also worked on something exciting about the hippocampus, and that's there in this paper, Space is a Latent Sequence. And if you are interested in hippocampus and how space gets represented, you might want to have a look at this paper. Thanks a lot for listening. I can take questions if there is time.
Thank you so much, Dileep. Thank you so much for your interesting talk. And yeah, now we have time for questions. So if you have a question, please use a raise hand button. We have plenty of time for questions. Any questions, please? Yes, Mariana, please go ahead.
Mariana. All right, I'm a colleague from Marina, but anyway, thank you very much for the very interesting talk. And I have just a very quick question. Since in our lab, we're also modeling like cortical canonical microcircuits, like I was wondering if you compared like to just like pattern of connectivity, did you take into account also, I don't know, homeostatic properties like synaptic scaling, also spike frequency adaptation, this kind of neural dynamic?
So yeah, so our model does not go into the level of spike dynamics. So you can think of our model as thinking of in terms of rates of firing of neurons. Yeah, so it doesn't go into the level of spike dynamics and probably not homeostasis. Yeah.
No. Okay, so rate based, you said everything. Thank you so much, and great work.
Thank you.
Another question from Craig.
Hello, thanks for the talk. I just wondered how you had a comparison between where your network tried to recognize characters. I mean, how does this compare against standard optical character recognition, such as Google as a Tesseract or the main system there?
Way better. Tesseract, standard OCR cannot solve CAPTCHAs. That's why CAPTCHAs existed. OCR existed before those text-based CAPTCHAs, and yeah, so the text-based CAPTCHAs that we were able to solve in our model, using our model, were not solvable using OCR before. And yeah, so they don't work well. And the reason why they don't work well is that in CAPTCHAs, in addition to these characters being distorted, they are also overlapping in complex ways. And OCR typically does a detection pass, which is separate from the recognition pass, and separating the detection and recognition is always problematic because when the scene is complex, you need to explain away things. As I said, so like if you run Tesseract on it, it might detect M as a solid evidence, and then it might, which is wrong when you look at the long context. So it's yeah. So unless you do segmentation and recognition in a unified way, there is no easy way to solve those CAPTCHAs. Yeah, so that was the point of the paper, showing that we can do way better than the standard OCR.
Okay, and so I had a student working on a project trying to look at old newspapers to try and find information about mines, and he didn't actually do much optical character recognition in the end. But I mean, is your network in a library he could have used in future?
Yes, there is. If you look for our just that paper, the RCN generative model Python code, you can there is a basic version of the model released open source, so you can test that. It just doesn't have the parsing aspect, which we did not manage to release at that time. And now the company is acquired, so I don't know where the code is. So, but at least the basic version, single-digit recognition is there, and then you can, it's not too hard to implement a parsing version based on that.
Okay, it's great. Thank you.
Thank you. Another question. Do you have questions? Okay, so I have a question actually concerning the RCN and the fact that it extracts features from the contours of whatever, like a capture or whatever object in front. But I'm actually wondering how it can go beyond this and try to model spatiotemporal relationships that might exist. Is it up to this level yet or not?
Yeah. So so we are already working on that one, and in fact, the earlier incarnations of this model also had the temporal aspect in it. It's just that during publishing, we just took it out to make it simpler. And the place where the temporal model fits in is in conjunction with pooling. So you can think of imagine a vertical line, and when objects move, that vertical line will translate to different locations in the neighborhood. And those will be learned as different sequences of that vertical line in our model. And then those sequences are then pooled over in the sense that you can think of these sequences forming a local network of this. This is how a vertical line can move to in the nearing temporal neighborhood. And then all those different motions of the vertical line are pooled as an invariant representation. This is basically saying different translations of the vertical line does not matter when you look from the next higher level. So pooling and temporal dynamics are very intimately related. The invariance is basically what we are assuming is that any invariance that we think about in the visual system is coming from learning the temporal dynamics. You're just trying to pool over variations that happen in local time. So in our model, as we are building now, we have these local dynamics being modeled as sequences, and it is very related to our hippocampus model in how the sequence modeling works in that locality. And then it's a community of that local sequences becomes a pool, which is like the convolutional pooling. So you can think of the convolutional pooling that is hard-coded in convolutional neural networks can actually be learned using temporal proximity. You are basically pooling over translations because translations occur in temporal proximity. And those internal dynamics of that translation is modeled within the region, but when you send the message out to the region, what you're sending is the invariant representation. That hopefully that roughly answers.
Yeah, and thank you so much. And does this actually valid as well if we speak about spatiotemporal relationships, like we were speaking about lines now? So if we speak about objects, can we also tell them?
Yes.
Okay.
So the higher levels of the network will then model spatiotemporal relations between objects. So so even how will they occlude in between, or how they may bounce, etc., or how they are. So that's exactly the idea. At the lowest level, it will model local temporal dynamics, but as you go up in the hierarchy, it will have long-term temporal dynamics of bigger pieces in the visual scene, which will be objects.
Are there any published work around this, or still underway?
It's still underway. Yeah. Hopefully in a few months.
In a few months. Okay. We have another question from Oksana. Oksana, please.
Hi, my name is Oksana. So I actually just wanted to jump on your last comment about the temporal relationship between different features and how it can, how can we derive the objectness from that? So that really kind of rings a bell for me in terms of sensory motor contingency theory.
Yeah.
I was wondering if you included it in your work or if you take inspiration from that theory.
I do take inspiration from the theory, and I do want to bring it into the work. So there are multiple places where it would come in. So one is so you can think of RCN as being a parallel model of objects. Basically, you present an object in, and without moving your eyes, you just do parallel inference, and some objects pop out. So it's a and if it's a video, you can just do again the same thing where you are not moving your eyes. It's just a video coming in, you're passively viewing it, and objects pop out at the top. Now, that's not the only thing that humans do. We also, when we are dealing with objects, we move our eyes, we walk around, all these things, and all those interactions also become part of our model, right? So it's not just what we see in a single frame. How then we can, if the RCN gives a parts of the scene, how do we attend to different parts of that object using eye movements or by moving around? All those become part of the model of the object, and those pieces can also be queried during inference. So we can do active inference on them, basically say, oh, RCN gives up one guess of what the object is, but now we can check because we have this sensory contingency model, and we can also query it both ways. You can do queries from the passive vision model to the active model and vice versa. In fact, both models are intimately connected. And it's very much also related to this hippocampus model that we have put out, the space is a latent sequence model. And you can think of that hippocampus model as a sensory contingency model, because all that it is modeling is how does the sensation change with respect to actions. And the idea of space is coming out fully as a, just a sensory model contingency. There is no Euclidean assumptions in that model. There is no location being sensed, and location emerges purely as a sensory motor contingency in that model. And the connection between that and RCN is that in that hippocampus paper, we are treating vision as a monolithic, a frame just treated as a monolithic thing without analyzing the contents. But when that hippocampus model connects to this RCN model, which we haven't done the connection yet, we are working on that, then it would be analyzing the contents of the visual scene and also then connect to the eye movement. So you will have a large sensory motor network with many contingencies encoded in the same network. So that's the way we are thinking about it.
Thank you. That's really, really exciting.
Thank you. Any other question, please? Any other questions, please? Okay, so if not, please join me to thank again Dr. Dileep George for his very interesting talk, inspiring talk. And thank you so much for being with us today. And the recording of this talk will be uploaded to the website of the CRNS talk series events webpage. And just to give you a hint, our next talk will be in May with Dr. Ali Agha from NASA JPL. So just please keep following our news. And thank you so much, and have good evening or afternoon or morning, depends where you are. Thank you, Dileep. Thank you, everyone.
Thank you. Bye-bye.
Bye-bye.